And now it's my task to introduce you to our next two incredible speakers. We're very blessed here in Michigan because we have such a richness of resources, and particularly in people who have dedicated their lives to epilepsy as physicians and researchers. So I'm going to introduce our two speakers. Dr. William Stacey is an epileptologist with the University of Michigan's Comprehensive Epilepsy Center. He conducts research there on physiological generators of seizures and novel device therapies. And also Dr. Wang, who's also an epileptologist at the University of Michigan's Comprehensive Epilepsy Center. He studies the genetics of epilepsy and how this can lead to therapies. This session is generously sponsored by the University of Michigan Health System. I know that our speakers have a lot of ground to cover, so I'm going to invite them to the stage for their topic, The Nature of Epilepsy Research Highlights. Dr. Stacey and Dr. Wang.
Well, thank you for inviting me to speak today. I'd like to first state a little bit of my background. So I am a neurologist specializing in epilepsy. I'm also a biomedical engineer. And that really stemmed from my interest both in devices and electronics and also in understanding the brain. I've decided to, as she just said, to dedicate my life to trying to understand and fix seizures. So as a physician, I want to understand the underlying mechanisms, and as an engineer, I want to fix it. And it's very touching and frustrating to me to see how difficult seizures are for people. I'm going to go through very quickly some things that, a little bit of background, because I want to talk today about some pretty in-depth things. We're going to be talking about the cutting edge of understanding seizure networks. As we all know, epilepsy sucks. And it affects 50 million people, 3 million people in the US, and unfortunately a third of them do not respond to medicines. That number, a third, has not changed since we started giving people medicines. And that's very frustrating. We started off with some early medicines that kind of hurt people. We found some other medicines that weren't so bad, but we don't like so much anymore. And we've made newer and newer medicines that are nicer and nicer side effect-wise. But that number of 30% has never changed. We can do a little bit, in some people, we can do a surgery. And if you're the right person with intractable epilepsy, we can take out the region that causes seizures, and those people usually do pretty well. But to be honest, we really just don't understand what causes seizures. And part of that is due to the technology that we have. If you look back, the early 20th century, we started giving people medicines. We then figured out that seizures were due to electricity in the brain and started looking at that electricity with EEGs. And here's a nice picture of a 1950s EEG. Some of you may remember those with the pens that shook back and forth and drew little lines on the page. The page went by. That device is very important, as I will talk about. We then developed different types of imaging that helped us see the brain a little bit better. We've now developed some devices that we can implant in people in addition to, or perhaps in lieu of, medications. But after all of those things, we still have 30% of people that don't get better with medicines or whatever treatment we have. And so we have been trying to make some devices. We have an amazing digital age where we can create things. These are all real pictures, by the way, real things. You can control video games with your mind. We have robotic hands, not quite up to Luke Skywalker level, but we do have, we're headed that way. And now we can wear things on our wrists that do so many different things. So why can't we do that type of technology to help people with seizures? And a lot of people have been trying to do that. So back in 2004, a newspaper published this joke. These are actually real patents from that year. And you got things such as a falling snowflake simulator and mosquito-killing bird bath, eyeglass wipers. And then if you look, one of the crazy patents they came up with was an implantable seizure sensor. That device, I'll talk about in a second, that's a real device. So the first one to come out was the vagus nerve stimulator. This is what's called open loop. You set it to a certain level, and it just turns on and it does its thing. We don't understand very well how it works. It does reduce seizures in people. It's not a cure-all. None of these things are cure-alls. They work about like another medicine. Now, the company that does this has now entered a clinical trial to change this a little bit. So one of the things that this device has is a magnet. Those of you that are familiar with it, you can swipe the magnet over your chest when you feel a seizure coming on, and that might help abort a seizure. And they've got a new device that would try to detect a seizure as it comes and then automatically do a magnet swipe. This is what's called a closed loop device. Now, I hate it when people have typos on their slides, and I do right here. I'm going to blame Russ Derry because between me writing the slide and it getting on this computer, it was in his hands. So it's not a seizure rat, it's a seizure rate. It's a heart rate. The next device to come out, this is the one with that crazy patent from 2004, the NeuroPace responsive neurostimulator. This is now available. It's FDA approved. And this works in a different fashion. So now we're going to put electrodes, rather than connecting it to the vagus nerve in the neck, we're going to put electrodes right on the surface of the brain. And instead of turning on all the time, this is going to sense and try to determine when a seizure starts, and then like a defibrillator in the heart, zap the brain and try to reset the brain and stop the seizure that way. Again, it's not a cure-all, and it works about like another medicine. Something in the pipeline, this was doing its clinical trials at the same time as the NeuroPace device. It actually went to the FDA first, and the FDA said no. Very close vote. In Europe, it was approved. A lot of people are hoping that there's more information that maybe this would get approved in the future. But this is the same device that people use for Parkinson's disease. This is an open loop stimulator. It just zaps every, you set the time and how fast you want it to go. It's placed deep into the brain, and it just does its thing. And it worked about like another medicine, but there were some concerns with it, and so the FDA didn't approve it originally. Now, it is used in Parkinson's disease all the time. It's just for epilepsy, they weren't quite convinced. Another thing in the pipeline, which you may not have heard of, is trigeminal stimulation. The trigeminal nerve is what innervates the sensation in all of your face. It's a massive nerve. There's all these things that happen in your face. And instead of implanting an electrode around your vagus nerve and sending it up, they had kind of the same idea. What if we try the same thing, sending it through the trigeminal nerve, and they can just put little tabs on the side of your face, buzz, and it sends a signal back into the brain that way. And it seems like it might be working. We do not have any data on this. This is still just in its clinical trials. So the data that we have been preliminary studies. I don't know if it works yet. And then another device that is very dear to my heart is a seizure prediction device. Now, we've learned how to predict things like the weather very well. We can predict things like earthquakes and avalanches fairly well too. We haven't been so good at predicting the stock market. So if you look at that, that tells you there are some things in nature that are predictable, but people are not. And what are seizures? Are they acting something under natural laws, or is it something random like a person? We're not quite sure. A company called NeuroVista decided to put millions of dollars into this question, and they developed a device, and they did a clinical trial in Australia on 13 people. They implanted this device. It recorded from their brain 24/7 for 13 months on every person, and they tried to figure out, is there some signal in the hour or 2 hours before a seizure that might tell this person, hey, you're about to have a seizure. Don't go anywhere right now, or take another medicine right now. And it worked in about half of the people. Those people loved it. And the other half, it didn't tell them anything, but that was really no different than the lives they were already living. Unfortunately, that company went bankrupt because it wasn't convincing enough. It was going to be too hard. They haven't totally given up on the idea. The company itself doesn't exist. But to me, this is fantastic because those 13 months of recordings from patients is a gold mine of data. And I am using this data right now. I'm collaborating with the people, and they're sharing the data. The people from Australia are sharing this data with everybody around the world who wants to work with it. So now I'm going to move on to new technology, some stuff that is in use and some stuff that's we're going to get more and more in the pipeline as the talk goes on. So first I'd like to talk a little bit about resolution. So you see here a picture of 4 different resolutions of photograph. We've got an old camera, a somewhat newer camera, a pretty good camera, and then a 4K camera. And I don't know if any of you have 4K TVs yet. I think they're too expensive for my blood, but the picture's really, really nice. The resolution is fantastic. And a resolution can talk about lots of different things. So for instance, the resolution of this meeting, the highest resolution would be the video that shows everything that is spoken, every single picture, every single second. If they just took a picture of every speaker and put up those 15 pictures, that would show you a little bit about what the meeting talked about, but not have a lot of information. If they picked 10 seconds from every talk, you would get a little more information, but you still wouldn't understand all the nuances of what are going on. That same thing happens when we record the brain in EEG. There is a trade-off. It would be great to record from every single cell, every single instant, but that's just not possible. And back in the 1950s, when they started doing this, there was a severe limitation. Those pens could only shake 100 times a second. The word hertz, HZ, means times per second. So if you've got something shaking 100 times a second, you can only resolve something that is less than that frequency. The problem is that nerves are a lot faster than that. So if you are sampling at 100 times a second, you are ignoring a lot of the information. That's like taking a 5-second snippet of every talk. In addition, when they first developed the electrodes to put on the brain, now I'm talking about the intracranial electrodes that we use during surgery, they're about 1 centimeter apart. It was just chosen at random. Now that again is giving you a spatial resolution that is very small. Now what they did in the 1950s was fine for almost every patient. There are certain seizures like this. This is an absent seizure. That's very obvious what it is. The EEG is perfectly able to tell us the answer. But what I'm talking about today are those people in whom we don't know the answer. So we've started to apply different types of technology to understand it. One thing that most centers now have is computer-aided EEG. So the EEG is here on the bottom, and on top, you see that the computer has done some processing to help us as clinicians find things that might be interesting. Other things that people are doing more on the research side is instead of using this type of electrode here, this is the normal one, it's about the size that when we implant an electrode, the relative size of it, that records from thousands and thousands of neurons at a time. But if you use a smaller electrode, what we call a microwire, it can record from a much smaller population of cells. Would that give us a better idea of what was going on? One example that I like to use is if we used one microphone in the middle of this room when everyone was chatting before the meeting, you wouldn't really be able to understand what was going on. But if you had a microphone over every table, we'd have a better idea. And a microphone in front of every person, we'd know exactly what was going on. But of course, a microphone in front of every person would be really hard to process. You'd have to listen to 500 different conversations over and over and over again. The sampling rate thing that I talked about, this is the range that they usually talk about. It's just under 1 time per second to about 30 times per second. But the range that neurons act on is this huge range that you see all the way from 0 to thousands. And people are now trying to sample at those different rates. So some things that we've done, these are called hybrid electrodes. So the normal electrodes that we implant in the brain are these big discs, and there are people that put microwires in between them, in the little spaces in between. This is extra information. We're not quite sure what to do with it yet, but it's being processed by many researchers around the world. People have developed even fancier electrodes. This is called the Utah array. As you see here, this is smaller than a penny. It's 1 millimeter. It's 4 millimeters by 4 millimeters. And it has 100 electrodes in it, and this thing's implanted into the brain. People have also developed some tiny electrodes using computer technology that these can conform to the surface of the brain. And our current state-of-the-art that is used at Michigan and several other places is that we are now using technologies like those to look at some things that people haven't ever seen before. And the one that I'll talk about the most today are called high-frequency oscillations, or HFOs. There's been a lot of research in things like this, but it's still in the research stage. And as is common, if you follow drug development or any type of treatment, what normally happens is somebody has a great idea, they publish a paper, and they say, look how awesome it is. And then when you start to try to apply it to the real population, it isn't usually as good as people say. So you have to be careful, both when you read things about medicines and about new technologies, because things that are reported are under perfect conditions, and they might throw out situations that they don't like. And as of now, this is still in the research age. And what these HFOs look like is just this buzz. That buzz is very, very short and very, very small, and you cannot see it on a normal EEG. You have to use special computer processing to be able to see this sort of thing. So let's talk about some of the different types of research that we have. The first is using this Utah array. A group at the University of Columbia decided to filter the data and look at just high frequencies. And what they found was this slowly moving wall of fast activity that crawled across this 4 millimeter space over the course of about 30 seconds. Now, the thing on the right is what you would have seen. It would have just looked like a big messy seizure with a bunch of spikes. But looking at a smaller resolution, they saw this tiny population of cells that seemed to be driving the whole thing. Now, those cells, this is 2 millimeters by half a millimeter wide. And their idea is perhaps that is the seizure focus. What would have happened if they had been able to resect just 2 millimeters of tissue? Would that have cured the seizure instead of having to take out a very large piece of the brain? They are thinking, this group believes that that is the true seizure focus. And that's something that we'd never seen before because we never had the resolution to see it. Another group at the Mayo Clinic uses those hybrid electrodes, the micros combined with the macros, and they found that if you look at just the microelectrodes, sometimes one of those wires will have a seizure and nothing else has a seizure. And their idea is that perhaps there might be little tiny islands of microseizures that you can't see with normal EEG, and those seizures might start 10 or 30 seconds before the real seizure. And then they grow and coalesce into a bigger island, and then once this big glob of cells, then it's a seizure you can see, but perhaps harder to stop. Maybe if we could look at that smaller scale, you might be able to buzz a tiny area of tissue and stop the seizure. Another idea, and this is a picture of real brain on the left here. It's a cat brain, actually. So they induced a seizure on this cat and used very high-resolution electrodes. And what would have looked like a normal seizure when you looked at every electrode, and what you see here in this movie is each electrode has its own pixel. Instead of being just a messy seizure, it actually was a spiral wave. So it was like a wave spinning around and around on the surface of the brain. And what's interesting is right there, the end of the seizure, that spiral wave went backwards. So this group is thinking that perhaps sometimes the seizures that we see, we look at them as a big glob of cells all firing, but in fact it has a very interesting topography underneath. And that maybe if you used some fancy technique like trying to induce that backwards spiral, you might be able to stop the seizure that way. Again, this is stuff we've never seen because we've never looked. Now, all that stuff I've talked about has been electricity, but that's not everything that's going on in the brain, and it turns out that if you look at other things like drug concentrations or the energy or the amount of oxygen or electrolytes like potassium, that's the K+, all of those things change during seizures. This is something that no one's really ever looked. So this is a group that I worked with in France, and they are very good at looking at what's called multimodalities. So instead of just electricity, we're going to look at a lot of things at once. And all of those things are changing, but we haven't quite figured out how to correlate all of that stuff. This is ongoing research. And then in the more theoretical side, we've partnered with a physicist who really likes looking at the dynamics, like how do populations decide to join together and do things all in a synchronous fashion, which is a great description of what neurons do during seizures. There are some very well-known among the physics community, some very well-known ways to describe that type of activity. And he is looking at how this can model human seizures. Now, it's interesting to model it from a theoretical point of view, but what I like about this idea is that there are some of those descriptions that will respond to one type of stimulus, and others descriptions describe to another type of stimulus. So what I would like to do with this is, can we look at an individual patient, see what their seizures do, and then figure out what the correct type of way to stop their seizure would be. That's, again, something we've never looked at because we haven't had the technology to do so. And then finally, these high-frequency oscillations. This is an example here on the top of what a normal EEG looks like that we as clinicians see. That if you take one of those electrodes, spread it way out, filter it, blow it up, and look at a little tiny piece, you'll see this high-frequency oscillation living on it. Now, these HFOs, an example of what the HFO looks like, this light blue line that's streaking across the screen is what the old EEG with pen and ink would look like. But if we sample faster, you can see there's a lot more stuff going on, this black line with all those squiggly lines. Well, those squiggly lines, those are the high-frequency oscillations. They are often a very good way to find where seizures are starting. The problem is they often happen in normal brain areas too. And so it's tricky to figure out when is one of these things indicating epilepsy versus when is it just indicating normal brain activity. And doing this type of processing is very, very time consuming. So as of now, most people that do the research, they have to manually go through, and that whole process of taking a piece, blowing it up, filtering it out, looking at it by eye, that takes hours and hours to do just 10 minutes of information. What we're doing at the University of Michigan now is trying to see what would happen if we introduced these HFOs into our clinical studies. It looks like they help us identify where seizures are coming from. So in a person where we can't find when and where their seizures start, maybe we could use this extra information to inform us, this is the seizure network, this is when seizures are getting worse, and then use that to perhaps help the patient in a way that we haven't been able to before. So our plan, first of all, is to figure out how to get clinicians to see the HFOs. What we do is every single patient that comes in for epilepsy surgery with implanted electrodes, we save their data at higher resolution. We now have a system that samples the data fast enough that we can see them. We save all of the data. We remove everything from the data that identifies them. So now it's just a bunch of random numbers. And we run an automated detector on it. So instead of taking hours and hours, we can do in real time, we can find these high-frequency oscillations. And then we have a method that automatically uses them to try to identify where the seizures start from. So here's a couple of examples. So this is a patient who's 24 years old, had horrible epilepsy, and we did everything we could to figure out where the seizures were coming from. This is something that our technicians are very good at the University of Michigan, is doing what's called an ictal SPECT. So we do an injection right as a seizure starts, and that shows us what part of the brain was starting the seizure. And his ictal SPECT showed bad, both sides. We don't like that, because we can't do surgery on both sides of the brain. So we brought him in for monitoring, and this is his brain with where the electrodes were, and we found that, sure enough, he had a bunch of spikes, which are the red dots on both sides, but it looked like the seizures were starting just in one spot. And he was able to have a resection of that one spot of his brain. This is a spot that we can take out, and people do just fine. So he had, for a couple weeks, he didn't feel so good, but after that, he was back to normal. We then looked at his high-frequency oscillations, saw kind of bad news. Now this was, we used him to figure out how to do this, so we had this information after the fact, several weeks later. It turned out that those high-frequency oscillations were everywhere, and unfortunately, the seizures did not get better. And that's kind of what this would have predicted. We needed something else to do to help him. Another patient, same thing, we put electrodes over a wide area, and the little oval-shaped things here, what we call mapping. So we had figured out what parts of her brain were important, what things were involved in language, motor, and sensory function. Those are areas that we would never touch. The other areas where the seizures were coming from actually weren't doing anything important, which is good. And her seizures all started on the bottom of her left temporal lobe, and sure enough, the high-frequency oscillations were also on the bottom of her left temporal lobe in the same area. Now, we would have had that information without waiting for a seizure. Interestingly, she had another area of high-frequency oscillations that you see back here in number 21. Now, that would seem like it's the wrong answer, but I know this patient, and that actually is an area where she also makes seizures. But those seizures she doesn't notice. So she ended up having a temporal lobectomy and has been seizure-free ever since. This is just showing us what our algorithm does. So it takes the raw data, and the computer goes through and finds these high-frequency oscillations. But what makes it really tricky is that we have a lot of artifacts. So every time the patient moves, it can make something that looks like a high-frequency oscillation. And that's what a real one looks like. Now we have this algorithm that identifies where we think the seizures are coming from, and in true University of Michigan fashion, blue and gold, or maize and blue, is the good, and the red is bad. So red would be a false alarm, and our algorithm, compared with some other things people have tried, was able to identify where the seizure started from, or identify that it couldn't figure it out. So the blue in this case is I can't figure it out. It's important not to make a bad decision when you can't make a good one. And so far we have collected, this is from a paper that we did, we've now collected 23 patients at the University of Michigan, and we have millions of saved HFOs. And that's something that we like to do. We are using a lot of high data processing, what's called machine learning, to look at these millions and millions of things and figuring out ways to try and identify what's normal and what's abnormal. Now, to do this, we've had a large group at the University of Michigan. Not everyone is on this picture, so I'd like to thank all of them. Also thank the funding that has helped this research go along, and thank the Epilepsy Foundation, who I definitely do endorse. So, thank you.
Thanks for the invitation, and it's my great pleasure to be here. And it's a very good place to meet my old friends and start making new friends. But first of all, I'm very nervous because first of all, Dr. Stacey just gave a wonderful talk, and I feel a lot of pressure, so thanks. The second of all, you are supposed to get Jack Parent, one of the best epilepsy researcher, to give this talk. but he is out of town and you get me. I'm not sure if I'm a good replacement. The last but not the least, as a physician and scientist, and the moral responsibility is to build a better world for our patient and children. So do we really understand an epilepsy? Can I offer a cure to my patient? So the nervousness and anxiety always comes from the very simple and basic question. So have we made a better world for our patient? So at this moment, my one side of the brain is telling me, yes, we have. So modern technology have significantly improved patient care. For example, with better MRI, higher resolution MRI, and better algorithm and protocol, we can see what we couldn't see with the past old MRI machine. So this is a patient of mine, 41-year-old gentleman present with multiple seizures every day. The old MRI didn't show anything, but the newer MRI at our institution with better algorithm clearly shows something in the left front lobe. Based on this finding, the patient underwent surgery and he became seizure-free. When the advanced MRI couldn't show anything, we have PET to show something abnormal based on the better understanding in the physiology of epilepsy. So the MRI didn't show anything. We sent the patient for PET, and the PET obviously shows something abnormal in the right temporal lobe. Based on this finding, the patient underwent surgery again and became seizure-free. And from last month, he started taking just one medication. In the past, he was taking 4 different medications and remained seizure-free. So when MRI and the PET scan failed to detect any seizure tissues, we have SPECT who can tell us where the seizure starts, spread, and ends. And our surgical techniques also keeps evolving. So from the basic intracranial grid implantation to the intracerebral depth implantation and stereo EEG implantation, we can sample all seizure activities in the seizure focus that are not accessible otherwise. And right now, let's switch the gear a little bit and take a look at the basic science. So from my perspective of view, we have 3 most pivotal breakthroughs in science in the past decades. So first of all, the Human Genome Project decode all human genome sequence, which is providing a roadmap for accurate diagnosis and a blueprint to design targeted treatment for genetic disease. And then we discovered a protein called Cas9, which is able to modify any genes so easily that every biomedical research lab right now is using this technology to study the mechanisms of disease. Oncologists have already created their own immune cells with Cas9 modified genes and use those immune cells to fight cancer cells. And finally, we have iPSC technology. So we can take our skin cells, grow them in the culture, transform them into stem cells, and then differentiate them into all types of cells in our body, including brain cells, heart cells, muscle cells, and kidney cells. So this technology makes regenerative medicine and tissue engineering just within our fingertips. So yes, we're doing a very good job, but then another side of my brain asked me to start to think about a little bit, is that really true? So 1 out of 26 people here will develop epilepsy in their lifetime, and we have no ways to prevent it. And for about 50% of people who have epilepsy, we don't know why we cannot give the clear explanation. And then no matter how many new medications are approved by FDA every year, still 30% of our patients cannot achieve seizure control. So this is not the problem only for epilepsy. 100 years ago, Dr. Alzheimer described the first patient with Alzheimer's disease. 100 years later, we don't have any symptomatic treatment, let alone for the disease-modifying treatment. The term, or the word autism has been used for 100 years, and today the treatment is very, very disappointing. And for epilepsy, we started using phenobarbital to treat seizures 100 years ago and dilantin 80 years ago. And right now, no other medication are better than these two medications regarding seizure control. So there is a huge gap between our powerful biomedical technology and insufficient knowledge and treatment for our patient. The problem for epilepsy is more severe. First of all, the epilepsy research is severely underfunded. So if you look at this number, based on the prevalence of each disease, epilepsy is the least funded research area. So stroke gets 2 times more funding than epilepsy. Alzheimer's disease gets 3 times more. Parkinson's disease gets 6 times more funding. MS, multiple sclerosis, gets 7 times more. And ALS gets 6 times more funding as compared to epilepsy. So it's very, very frustrating. So anyway, so how to bridge the gap? I think the first thing is to increase the awareness in our community, in the private sectors, and in our government. And we have to invest more to understand epilepsy in order to help our patient. And I just came back from a stereo EEG training course held in Montreal, Canada. And it was called North America Training Course. But all the speakers came from Italy, France, and Canada. It's shocking how far behind in knowledge and technology for epilepsy in the United States. So, and because of lack of funding, we are losing many, many bright and smart young people, and we have to encourage medical students and researchers to join our team. And the last, we have to put more emphasis on translational research, which is bringing the powerful technology to the bedside patient care. So let me just give you a very short story about how we do the translational research in our lab. So it all started with a phone call from my collaborator asking for help. So he had a 1-year-old boy who presented with frequent seizures and some developmental delay. So then he sent me the EEG, which showed disorganized background and very frequent spikes and sharps in multiple regions. He showed me his MRI, which clearly showed atrophy in the brain, particularly in the cerebellum and the brainstem. No specific diagnosis was given to the family, and the boy didn't respond to treatment at all. So I'm not a pediatrician, but I believe this is not uncommon situation in our daily practice. So no, very, very sad story, no diagnosis, no treatment. The family is left in despair, and they start blaming on themselves. So we have powerful technologies. powerful sequencing machine, the Cas9 technology, and iPSC platform. So what does this child have? Why this child has epilepsy? And what is the treatment? Can we use this technology to answer these questions? So first of all, what does this child have? Does this child have genetic mutations? So we drew blood and send the blood to do a whole exome sequencing. And an extra non-base pair duplication in SP10-1 gene was identified. And SP10-1 is coding for a protein called alpha-2 spectrum, which is localized just underneath the cell membrane. It appears that this protein is very important in neuronal function. So we have a diagnosis, but we don't know why this genetic mutation causes epilepsy. Without knowing the mechanism of this genetic mutation in disease, there's no way to design an efficient therapy. So then we use the Cas9 gene editing method to study the mechanism of this disease. These are normal cells. So we can see these cells or neurons have nice axon and a very beautiful tree-like dendritic architecture. And these cells are SP10-1 mutation cells that are created by Cas9 technology. So clearly, those cells don't have axons and don't have the beautiful tree-like dendritic architectures. And we also studied the electric activities on those cells. And you can see there is a huge difference. So we conclude the lack of inhibitory activities on these cells lead to the frequent seizures. So we have diagnosis. We kind of know why this boy have seizures based on the mechanistic studies. So what's the treatment? So the boy already failed all medications on the market, suggesting that his unique genetic mutation and his genetic background may require individualized precision medicine rather than one-size-fits-all medications, anti-seizure medications on the market. So how to design individualized medication? First of all, we have to get the brain cells from this boy and test a large number of drugs or chemical compounds on those cells. So the first task is to how to get the brain cells. The second question is how to do the drug screening. So fortunately, we have iPSC technology, so we don't need to take the tissues from the brain. So we took brain skin cells from the boy and culture them in the petri dish and transform them into the stem cell. So the figure A is skin cells from this boy. Fig B to fig D are the cells that have been transformed into stem cells. And from fig E to fig L are the stem cells with some markers indicating those cells have potential to differentiate into all type of cells in the body, including brain cells. So then we differentiate those stem cells into neurons. So you can see on the very right side, this cells is like a neuron having the very beautiful tree-like dendritic architectures. So we have we got the brain cells from the skin cells from this boy, and how do we do the drug screening? So we use a 96 well plate. So we culture the brain cells in each small well in this plate, and each of these small well is also coated with tiny electrodes that are continuously recording the cell's activities. Then we apply different drugs or chemical compounds into this well and see if these drugs can change the cell's activity. So if a drug is effective in controlling the seizures, so the recording will show the activity will be decreased. So the decreased activity will be automatically detected by a powerful software. So the last question is, did we find a magic pill for this patient? The answer is no. The boy passed away, sadly, the boy passed away at the age of 3. But we are very proud that we are close, and we are doing our best to help the family, help the patient, and trying to build a better world for our patient. So the conclusion, so we have powerful tools to understand the disease and epilepsy and to develop novel therapies for our patient. There is urgent need to bridge the gap between the basic science and our clinical practice. And there is urgent need to develop precision medicine for our patients. The last slide is acknowledgement. So thanks for our patient who put trust in us and allow me to use their data. And thanks for Jack Parent, and he's my mentor, and the wonderful support from him, and our collaborators and advisors. And the project is supported by NIH and the Perf Foundation. Thank you.
Thank you very much. This is outstanding work. Question I have is really more about we all know that epilepsy is underfunded relative to other disorders, and so how do we change that? Number one, in your perception, but number two, what's the funding landscape look like? How do you have to go out and get funding, and who's funding? Is it only NIH, or are universities doing this, or other private organizations like NeuroVista that unfortunately are gone? How many companies out there are doing this type of stuff, and how do you advance that funding to be more in line with what we want?
Is this on? Now it's on. Primarily, it's through NIH. The NIH has 2 sections that are dedicated to epilepsy, although we share with stroke and some other things. We share the funding with them. So by far, it's NIH. The next, there are some foundations that help. The American Epilepsy Society and Epilepsy Foundation both sometimes fund, usually they're funding junior investigators for a year or so. But really the biggest foundation that helps epilepsy is called the CURE Foundation, cureepilepsy.org. And they have some major grants that are quite competitive. So that is, in this country, the best foundation. It's not the best, it's the one that has the most money to fund. And they have funded some very interesting, very prominent projects. Sometimes companies partner with clinician investigators to do different things. That is sometimes a little tricky for us because as soon as we do that, then we start getting conflict of interest problems. But that's it. You just have to be careful how you do it. But there are a lot of companies that do that sort of research. And then finally, there are a lot of private donors that fund as well. And so many different universities will have funds in memory of somebody, or just philanthropic groups that have decided to donate to certain things. Now, as far as the landscape goes, the funding is difficult. It's difficult in every disease. They take about, at the NIH, they take 15%, 12 to 15% of the applicants every time. So if you're not in the top 15%, then you're not getting funded. And it can be very frustrating. And a lot of people, and that's not just epilepsy, it's kind of across the board. So the question is, if in the United States, are we ahead or behind with compared to other countries? I think in most respects, the U.S. leads research around the world in everything. So our country dedicates more money to research than any other country that I know of. So other countries do have money. The European Union has done some big things, but they typically have a lot of money for a very small group of researchers. In the U.S., we try to do so many different things that our budget is much higher than any other place.
Hi. Thank you for coming in to talk to us today. I actually have 4 questions, and I know that we don't have much time, so if you can address a couple and talk to me after, that would be great. They're kind of all over the board. I was wondering what chromosome the SP10-1 gene is located on. Uh-oh.
I'm doing a lot of research on SP10-1 gene, but I forgot.
All right.
I think it's chromosome 21.
21?
Yeah, if I remember correctly. But it's easy to check.
Okay.
Yeah, I forgot.
Okay, okay. I was also wondering, you know, we have neurologist and engineer here, right? Neurology, engineering, medical doctors, but do you see any research coming out about metabolism? Specifically, why is the ketogenic diet, why does it work for certain patients? Why do these ketones, how can they stop the misfiring of neurons? Wouldn't it be interesting if we had neurologists and maybe physiologists who specialize in metabolism doing some research studies as well? Maybe that's going on. I just wondered if you knew.
There are people from both sides. It's very intriguing. We've known about the ketogenic diet for years, and they've tried it from both sides. They're still trying to, actually, so I have a student in my lab that was given a test question, and that was the question they gave her, because it's still one of the great unanswered questions.
Right, right.
I have a comment on the.
Oh, I'm sorry.
So there is a study on why ketogenic diet reduce seizure activity, and they link the mTOR pathway to the ketogenic diet. They found out the ketogenic diet can inhibit the mTOR pathway. That's why they say, because mTOR pathway hyperactivation is linked to the seizures.
The external stimulators, the stimulators that you talked about, I'm aware of, but I'm wondering if you envision a time in the future where perhaps we won't have to have an implantable stimulator in the brain, but actually something that's an external device, almost something similar to a hearing aid. Do you think that that's a possibility?
Well, there are other types of research I didn't talk about where you can send what's transcranial magnetic stimulation or direct current stimulation without breaking the skin. Those might work too. There are trade-offs, and the trick is to get the treatment to where it's needed. And for hearing aid, it's easy because it's needed right there, but for seizure, it can be harder to do that.
Yes, so in our lab, we're in the process of differentiating the stem cells into interneurons. But transplantation, I think we haven't tried yet. But other labs, such as Scott Barabin at San Francisco, University of California, San Francisco, use the interneurons from the rodent and transplant them into the rodent with epilepsy. And he observed the transplantation reduce the seizure activity dramatically.
Okay. Well, I think that's all the time we have right now. I want to thank Dr. Stacey and Dr. Wang. I think this has been really very interesting. I know there's a lot of questions unanswered here. We have gifts for you. Little gifts, just to thank you for coming today and sharing with us. We really appreciate it. We're going to take a quick break for 10 minutes. Please don't all mob them at the same time. But we would like everybody to come back by 11:10, and so we can start our next session. Thanks.